Okay. So essentially, the lecture exams, I have it written down here. So those days will probably be sticking to the review unless Dr. Osuna says anything otherwise, okay? So requirements of discussion. So for my, the sections include 25 points out of your 230 points in genetics course itself. You will be graded based on 3 criteria. This is, okay, so one of the things I should tell you, I am deviating a little bit from Dr. Osuna's syllabus on that part, where you end up with quizzes 10 points, attendance and participation 10 points, and presentations 5 points. So quizzes, it will be every week unless I say otherwise. They will be based on material that we cover in discussion or the, okay, not really homework, but the video, I'll be putting up a discussion video so you guys can watch that and answer it. So it's 12 quizzes in total. The final, the highest 10 will be counted into your grade. And there will be 1 point per quiz. It will be on Blackboard. The passwords will be within the discussion videos. Attendance and participation. So the way I'm going to be running these sections, if we stick to this plan that we have, the way I'm going to be running these sessions will be as a Q&A. So we're going to go through some problems and the student presentations that we need to do and cover anything else that anyone needs. And again, 12 sessions in total. You're allowed to miss up to 2 because I'll be counting 10 scores tops, okay? So presentations, this is a university requirement. So I cannot give you a grade in discussion without you guys doing the presentation. So please do. So the presentation is basically on a question that I've assigned you guys already. It should be on Blackboard. What the question is, if anyone has a question on it, let me know. It's 5 points based on, with a PowerPoint, where you have the question as one slide. Like you have the question, you give the background information to it. Like, don't just tell me the right answer. You have to tell me why that's the answer, okay? So an example would be this guy. It's not one of the questions that I use on the presentations. However, this is the kind of an example that I'm looking for. So which nitrogenous base is shown here? So we have these guys here. Okay, so we know nitrogenous base, right? So in this case, what do we know? We see a double ringed structure, which means purine, right? So as we look at this, okay, so once we see purine, we cancel out cytosine, thymine, and uracil, because those guys are pyrimidines, which are single structured. And then you go into the differences between adenine and guanine. With guanine, you should already know for a fact that you'll only have you'll have a carboxyl group up here. So, and this amino group is up here, so that should cancel out guanine, which gives you adenine. So the correct answer would be adenine. So does that make sense in how I'm looking for in your guys' presentation? Great. So the way I've been structuring my discussion section, so the purpose for discussion is to supplement the lecture, not do it all over again, because that would just be too much in the time that we have. So the way I'm going to structure this is that I'll put up some discussion videos. They will focus in on the concepts that I want you guys to focus on for my part of the course. But these are not comprehensive. So do not use these as a substitute for lecture. I'm sorry, I'm getting an email from a student. Hold on, sorry. Okay. Whoops. Hold on 1 second, sorry about that. Okay. Forgive me. Bear with me with this. It's been a little bit wild. Okay, let me just send one of the okay, sorry about that. Oh my lord. Online teaching has been a wild life lately. Okay. Sorry about that. Okay. So again, not a substitute for lectures. So make sure you guys do listen to those videos because I kind of cherry pick the major concepts that he has in there. So the quizzes will be connected with these videos. They'll be available Wednesday evenings and should be completed before the next session. The password to the quiz will be provided within these videos. So that's something you would do before discussion session, or in this case, after, okay? So it'll be assigned today. You guys will be able to do it anytime during the week. You can let me know if you guys have any questions regarding the material itself. And then we'll come into a Zoom Q&A session where that's where the attendance points will be and where we will do an activity or like answer questions, you know, like learn fun stuff about genetics. But as I was planning this out, I was kind of wanting to get your guys' opinions on how you guys would like this discussion to run. So one of the options that I have is the synchronous version, which is what I explained so far. Asynchronous is similar, except not all of us have to come in during the sessions. Only the presenters will be coming in during those sessions. And in this case, I cannot put in attendance points just because it will have to revert back to the 20 points in quizzes, as the lecture syllabus says. Then there's the self-paced options where we don't meet weekly, So we cannot do attendance points. Quizzes will be weekly, but you can do it until the time the lecture module exam comes in. Okay, so if you have a quiz and you do it by like if you do it weekly, it's better just because you're keeping up with the material and going through it as we go along. But as long as you have it within by the exam lecture exam, you guys will get credit for it. In self-paced, the presentations will be self-recorded. So that is one other thing to think about. I will, so for answering this, I will let you guys do a little form that I have set up for you guys. I'm also counting this as your attendance. So please do that. Like, I'll give you a couple of minutes to do that. Let me just find the chat box again. Okay. Okay. Yeah, maybe. The little box is playing hide and seek with me. Okay. 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 Let me just stop the sharing for a second. Oh, okay. Can everyone see the link that I just sent?
Yeah.
Can everyone open it and just fill that out for me, please? In the meantime, do you guys have any questions about like the structure of the course itself? Or did I just overload you guys with the nuances of genetics discussion? Oh yeah, and with either asynchronous or self-paced, I will definitely keep the sessions where I say exam review. So you guys can come in, ask any questions, or like we can go through much more problems and be all ready. But the one thing about that is that I don't see the exam until you guys see it too. So it cannot be completely the same, like, or similar to the exam itself, but we'll go through some problems that will get you prepared for the exam itself. Like get familiar with the concepts and the applications and whatnot. Do you guys have any questions or concerns?
I was wondering if majority of the class would prefer asynchronous. Can the people who prefer synchronous still have a synchronous class?
I'm trying to keep it whatever the majority says. However, you are welcome to come to the sessions where the presenters are coming in. It'll be mostly on the presentation problems itself. But if you guys have any questions, we can go over that too. So if it's asynchronous or self-paced and you guys still want to be synchronous with me, you can still do that. You can come in again. If we go asynchronous, we can definitely do the weekly meetings. You guys are welcome to join in any of the sessions, as long as you keep up with the quizzes and whatever else is there. Does that make sense? Did I answer? If we go self-paced, that will depend, because I do have the videos that I'll put up with these questions. So it'll kind of give you a sense of it, but I will definitely keep some sessions open to like my these sessions will end up turning into like a office hour per se. Like if you guys want a one-on-one meeting or that kind of thing, we can do that too with self-paced. Any other questions?
There's one in chat.
Oh, that chat box keeps disappearing on me. Thank you. So do all of the sessions have the same quizzes, just in case I want to join a session that isn't mine? Okay, so I keep it similar with all of my sections, so it's identical. I try to keep it identical at least, because I'm trying to go with the majority of my sections. So the quizzes themselves are on Blackboard, so you'll be able to do them anytime during the week. It's not during the session itself. Does that answer your question?
Yeah, answer my question.
Great, okay. But if you do want, if we do end up doing like the synchronous version, then you guys are welcome to come to any of my sessions. Just let me know beforehand if you can't make it to your own session. And also, I forgot to tell this to my 4:30 class, but if you can't reach me during my office hours, you are always welcome to go into the other TAs office hours. They're always happy to answer any questions. If it's section related, definitely come to me. That would just get more confusing for the other TAs. If it's material wise, you can go to them if it doesn't make sense from what I'm saying. Yeah. Any other questions? Okay, I see all of you got a submission in. Oh, wow. Okay. I guess my next class will be a tiebreaker. Okay. Okay. So I will definitely let you guys know which one will happen between the three different structures, just because I want to keep it identical between my sections. But again, let me know if you guys have any concerns about any of this stuff. I'm always open to tailor it again. Can you clarify your question, Rishabh?
I was just saying, like, what is it leading towards, like, self-paced, asynchronous, or synchronous?
Okay, right now it's tied between synchronous and asynchronous. So it's like 35% each for those two, and then 30% for self-paced. But I will definitely let you guys know which one will end up happening. I'd have to check with my third class as well, because I want to make sure that we're all like on the same page, I guess. But if it's still a tie, I will definitely make a poll again, just to see where you guys are still standing with that. Because it's literally split by there. Any other questions? I think I just had a student join. So questions wise in the quizzes, it's about 3 to 4 multiple choice, nothing too crazy, whatever we covered. Okay. Wait, I think we just had a student join us. So Quentin, can you see the link that is in the group, the chat?
No, I can't see it.
Okay, I'm gonna put that in again, because sometimes you cannot see it until.
All right.
So can you please fill that form for me?
Yep.
Did everyone else complete that? Yes? Okay. Okay. So do you guys have any questions? Anything else? Any concerns? Good? Okay. Well, that was my side. Like my little nematodes. Okay. So we're gonna go through a couple of problems that are relating to DNA structure and organization. I don't know how much we'll get through of this, but we'll get through as much as we can. So what are these nitrogenous bases? If you're saying something, Elizabeth, you're muted. Nobody likes nitrogenous bases. Oh, whoa, okay, chat. Why does this chat keep? Okay, I guess now my question is, which one is what are you guys thinking? Left is
Do
adenine,
we-
yes. Oh,
Do we just say the answer, or do you want us to put it in the chat?
you can just say it.
Oh, I think the one on the left is that adenine,
This guy? Yep.
and the other one is guanine.
Mm-hmm. So, like how I want you guys to do the presentations. Why? Why are those two the way they are? Like, well, I guess my question is, why did you say this is adenine and this is guanine?
Can I answer it too?
Yes, anyone can
Okay.
answer.
Okay, so at least the way that I have it is that for adenine, at the 6' it's the NH,
Mhm.
and then for guanine it's at the 6' it's the oxygen.
Mm-hmm. So obviously we know that these two are double ringed, so purine cancels out all three of the other sections. And then at that, this adenine has a amino group, which leads to adenine. Don't be fooled by this one down here, because the carboxyl group is up here. So that's what gives away guanine. So that's these two. What about these guys? You can just shout it out.
Is the one to the far left cytosine?
Yes. Why?
Okay. Because there's it's how do you pronounce that?
Amino?
Is that how you pronounce it? I'm not sure.
Yes, the amino group.
Okay, yeah, that one.
Yes. Or the NH2 group. As long as you recognize this and say it's single ringed and then NH2, automatically think cytosine. What about this guy? The one in the middle.
Is that one thymine?
Yes.
Because it
Why?
has the CH3 at the top left.
Mm-hmm. So this line here, it is a methyl group, a CH3. So that gives it away for thymine. And what does that leave us with here?
Uracil?
Yep. So these two are similar. As long as you remember thymine has the methyl group, you'll be able to distinguish those guys. Does that help? Okay. So that's uracil. Happy uracil. Okay. If an organism has 10% adenine in their genome, what is the percent makeup of other bases? How do you think we'll go about this?
I think I remember in the reading saying the guanine and cytosines are a third or half. I think it's a third of the percentage of base pairs, and I can't remember what the percentage was. I might be.
Wait, can I answer?
Yeah.
Well, if adenine is 10%, thymine would also be 10%, and then whatever portion is left, so that's 80%, your cytosine and your guanine would be 40 and 40.
Okay, so do you remember which rule it was that made that happen?
No, I don't remember.
Okay, don't worry. That's why we're here. So anybody else have an idea?
Wait, is the rule the base pairing complementarity or no?
Yes, and it's also called Shargaff's rule. So remember that guy? He said that they were all proportional. Adenine is proportional to thymine, cytosine is proportional to guanine, and that's how they ended up figuring out that they both pair with each other. So in this case, we, like Christine said, we have 10% adenine, so it ends up with 10% thymine. And then the remaining is about 80%, which is essentially how you would do that. And you get 40 for cytosine and 40 for guanine. Does that make sense? My phone needs to go away. Okay, sorry about that. So are we good with Shargaff's rule and all that fun stuff? Okay. I really need to fix this question. Okay. No. Go back. Okay. DNA and RNA are chemically similar and are distinguished in large part by the two prime area of it. In 2' okay, this is supposed to be 2' H hydroxyl group. I forgot the hydroxyl part in RNA, and 2' H group in DNA. Why do you suppose that both of 2' hydroxyl group that presence in group DNA and RNA have, oh my Lord, this question. Okay. So essentially this question, what it's trying to ask is, why do you think that the hydroxyl group in the 3' carbon, why is it that we have that hydroxyl group on 3' carbon and not find 3' hydrogens? Does that question make sense? Okay, I think I just confused everyone. Okay, so the question here is asking, why do we see a 3' hydroxyl group instead of a 3' hydrogen for these guys, for RNA and DNA?
Okay. Is it because I think it's because there's 2 sites for the adenine and thymine, and then there's the 3 sites for the guanine and cytosine, or however.
Okay.
Am I wrong?
So we're talking about the sugars, right? Ribose and deoxyribose. So on the sugar, in ribose, you have the 2' and 3' having hydroxyl groups. With deoxyribose, it's the 2' hydroxyl group in RNA that gets replaced with a, so you don't have a oxygen in the 2' carbon. So with that, why do we never see a 3' hydrogen group instead? Do you guys know? Okay. I don't know if this question was a little bit too confusing, but essentially, so, okay. I think the picture might help more. So this is an RNA chain. So you have 3' hydroxyl group, 2' hydroxyl group, right? When you see these guys, what the difference between ribose and deoxyribose is the 2' hydroxyl group. You don't have it in deoxyribose here. So the issue is that we don't, so it's asking why do we always see a hydroxyl group here instead of a 3' hydrogen? So why do we have this oxygen here? Because it's involved in making phosphodiester bonds. And without these phosphodiester bonds, you cannot have them becoming polymers. So you cannot create the chain of DNA or RNA at that point. Does that question make sense now? I promise I'll reword that when I put it up. We cool with this question? Okay.
Can you please, I'm sorry, can you please explain your answer one more time? I almost got it, but barely.
Okay, where did I lose you? That's my,
After you told me that it was the phosphodiester bond, they don't make, and then I forgot the term that you used, and then from that point on, I was gone.
okay, so here, the 3' hydroxyl group is the one that is involved in making these phosphodiester bonds. When you can't form the phosphodiester bonds, you cannot form the sequence or the polymer of DNA or RNA. We good? Okay. So what did Griffith do?
The transforming principle.
Mm-hmm. How did he get there? Any ideas?
He injected mice with a live strain of a viral bacterial thing, and then that doesn't cause death, and then he injected it with a heat-killed version that does cause death if alive, but and the mouse ended up dying only when you combine the two, because it was able to pick up the DNA from the heat-killed one, and the living one was able to use that to transform into the kind that kills the mouse.
Good. Okay, so essentially that is what happened. Griffith took 2 strains of Diplococcus pneumoniae, which is a bacteria that can be either virulent or avirulent. So the S strain, he took the S strain, put it into the mouse died. He, so these are his controls where he did, he took the R strain, checked the mouse, inject the mouse, and then the mouse lived. So these are their 2 controls. Next thing he did was he took this S strain and heat killed them, and he saw that the mouse lived. But the next experiment that he did, he took the R strain and the heat-killed version of the S strain. They put it into the mouse, and the mouse died. They were like, what? And when they saw that the tissue that they had, they were like, they had living virulent strains going on, and they were like, what is going on? So there is something like transforming, but at that point they didn't know what it was yet. So essentially it was asking whether there was a transforming factor or not, like you guys said. So wait, does that make sense? Okay. So the next question, the Griffith experiment, showed that certain strains of Streptococcus bacteria could transform from one strain to another when combined. How did Avery, McLeod, and McCarty experiment expand on Frederick Griffith's observations?
Is it A?
Okay, why? Again, like with the way I'm doing presentations, you guys have to remember, I like the answer of why too. Like, why is that the answer?
Can I explain?
Yes.
Okay. Well, they used like several proteins, and they first checked whether RNA was the transforming principle, and then they also used. Sorry, I'm looking through my notes.
I have the figure up here if you want.
Yeah, first they tested to see if it was. Sorry.
It's okay.
Yeah. They used protease.
Protease, yep.
Protease, and they canceled basically all of them out until they got to deoxyribonuclease, and they realized that that was the transforming principle. Sorry, that was a really bad explanation.
That's okay. It's okay. So do you guys know what protease, ribonuclease, and deoxyribonuclease does?
It cuts up the molecules so that they're dysfunctional.
So essentially they're breaking them down and making them non-functional. So these are enzymes that break and digest them down. So they kind of did like a process of elimination here. So first they took all the carbohydrates, lipids, proteins, and put it into the filtrate with the R strains. And they did that. They were like, okay, transformation is occurring. So there is the active factor in there. So next they checked if it was protein. When they did, they digested it with protease, and they saw that a transformation still occurred. So they concluded that the active factor was not protein. Then they tried ribonuclease, which breaks down RNA, right? So they took those and they saw that a transformation occurred again, and then concluded it's not RNA. Then they used deoxyribonuclease, and then they're like, oh, no transformation. DNA must be the factor that's involved. Does that make sense? So that is how our answer here becomes A. Are we cool with these two experiments so far? Okay. Next, this question is, oh, wait. Okay. A famous experiment demonstrated that DNA could direct viral replication, providing evidence that DNA is the genetic material. Which scientists were directly involved with this experiment?
Is it D?
Yes. So again, we talked about these two. They worked with Diplococcus pneumonia. Watson and Crick, they were involving the structure of DNA. So it leads to Hershey and Chase, which this is pretty much that experiment. Can anyone explain to me what's going on here? I promise I'll let you go soon.
I believe it's oh dang it. I know it, but I can't.
Elizabeth, you're muted again.
They're taking the DNA from the virus and they're extracting just the DNA, right? And then that's what the whole centrifuge process is. Or is that the last experiment? Is that this one? I remember they were taking the direct DNA and then they were, I don't remember, never mind. Bye.
Okay, so Hershey and Chase is the one that they used radioactive labeling in DNA, which incorporates P32 or the phosphate, and S35 for protein, right? So you come down, it goes in, both of them are labeled. They infected the bacteria, and after they infected it, they put it into a blender and went out. And they were like, okay. And then it came down, things started lighting up like Christmas trees. So in this case, in the DNA part, that's where the bacteria lit up. So one of the things that they were trying to notice is which one actually gets transferred into the bacterium itself. So they saw that the bacteria here were radioactively labeled when it was the bacteria. And this guy ended up with the phages, and this did not have anything labeled. Does that make sense?
I have a question.
Yes.
Now, what are the ghosts? Where it says like, you know, the phage ghosts?
Okay. Those are essentially the I guess it's another way of saying the phage without the genetic material. It's just saying that so at this point these phages cannot operate because they already gave in their genetic material, but they're still highlighted with protein and whatnot.
Thank you.
Yep. Okay. This is a very simple question, and I promise I'll let you guys leave after this one. How do bacteria package their DNA? Supercoiling, okay? It's just making it more compact, coiling it up, and be like make that big size of DNA into that tiny little cell by supercoiling and making it more compact. Does that make sense? Any questions, concerns? Okay. As of right now, okay. So that is all I have for today. If you guys, so I will let you guys know what structure we will take for the upcoming discussion sections. So let me know if you guys have any questions about the presentation, as we start those next week. So let me know. Okay?
I do have a question.
Yes.
I'm the question I was gonna ask, how are you gonna notify us about how we're gonna start discussions from this point on? Is it gonna be by email or by Blackboard?
Email.
Okay.
Well, I'll put an announcement as on Blackboard, but it will also send out that email with it.
All right, thank you.
Yep. Any other questions, concerns? Don't be afraid to bug me. It's my job. So, and I
There's
do.
one chat. It's asking if the quiz is on Blackboard.
I will do that today, but yes, it will be on Blackboard tonight. I just want to make sure everyone is, yes, there is a quiz this week. So as I said with the schedule, it's the date that I will assign it. So we will definitely have one. I will put it later tonight and you guys will be able to complete it anytime during the week before next week. Like even if we switch to asynchronous, we're gonna stick to the Wednesday deadline.
It's cold.
So we're gonna stick to that deadline, if anything. Anybody else, anything? If not, you guys are free to leave.
Thank you. Have a good night.
You too.
Thanks.
Tahia?